I would like to congratulate the founders of this beautiful institute, NeuroPSI. And yeah, also very pleased to see that so much exciting neuroscience is happening in Paris. And um, what um, Alain just said is true. So I have a dual appointment now. So I'm the director of the Netherlands Institute for Neuroscience, but together with people at the Institut de la Vision, we started an endeavor to also have what they call a non-human primate platform at CUA in Fontenay-aux-Roses, so not so far from here. And I bet that there will be many exciting opportunities for collaboration. So very excited to be here. So today I would like to talk about Stan's topic, conscious visual perception. So I'll admit that I have tried to avoid these very complex psychological concepts in my work. So I wanted to do something real, something that you can measure. And in the beginning, I said to myself and others, I'm not going to talk about attention because attention is so diffuse. Different people mean different things with attention. So I tried to avoid it, but at some point it became impossible to avoid it. So I had to talk about attention because there's so much psychological work that relates to the experiments that I was doing. It just became completely silly not to talk about it. And now the same happened again for the C word, C for consciousness. Okay, so I also now started to work on consciousness, and that's even more hopeless, right? Because the psychologists and the philosophers of consciousness, I mean, they're a hopeless bunch. They talk about concepts all the time, and that makes the whole field also a bit hard to work in. But yeah, I mean, I still, it's very important that this work is being done. And I try to focus on the data and not so much about the theoretical debates about concepts and see how far we get with this. So I'd like to make a small disclosure. I'm also a co-founder of a company, Phoenix, and in the last part of my talk, I'll be talking about a little bit about our endeavors to create a visual prosthesis for blind people, and that is related to this startup company. So today, I first would like to talk a little bit about feed-forward and feedback processing in the brain. That probably will last only 5 minutes or so. And then I'll be talking about visual awareness, the neural mechanisms that support it. And say the last 10 minutes or so, I'll dedicate to the work on visual prosthesis. So let's start with the introduction. So if you see this picture, there's a lot of information here. And you see something that is actually seems at introspection quite integrated, right? So if you look at the nose of this animal, you don't have a hard time seeing that it's the same animal as these legs. Now, that seems intuitively immediate, but in the brain, that's actually quite a challenge because you should realize that in the visual system, analysis starts with tremendous fragmentation. So neurons at early levels in the retina and the early levels of the visual system, like primary visual cortex, they have these very small receptive fields here indicated as circles. And at first sight, they only seem to be carrying what's going on in this very tiny fragment of visual space, right? That's not how we perceive. We perceive these animals that have a spatial extent. So there must be some powerful mechanisms in our brain that puts the fragments together again into wholes. And that is something that I've always been very interested in. Turns out that in the brain, this is solved by what one might call a labeling operation, where all those image elements that belong to a single perceptual object are labeled, and they're labeled with enhanced neural activity. I used to think that it was synchronization, but it turns out that synchronization doesn't play any role. It's really labeling with enhanced neural firing rates. So this is the human visual system. And as a model system, we use the macaque monkey, also the mouse, but I'll mainly be talking about the macaque monkey. And this is from a famous paper more than 30 years old now, where you see all these areas involved in macaque vision in different colors. And from the same paper, you can make this hierarchical scheme. And this is actually something that is quite well modeled by convolutional neural networks. And that is that Low-level areas are tuned to simple features, and then areas at intermediate levels feature constellations of intermediate complexity, and at higher levels you have neurons tuned to object categories. So that is the first feed-forward phase of information processing as the information goes from the retina to the higher cortical areas. But there are many recurrent connections, so feedback connections and lateral connections that allow for recirculation of activity. And what we find is that these recurrent connections are actually very important for putting the fragments together into a coherent role. So this labeling process that I was talking about, and I'll show just a little bit of data about that later during my talk. Okay, so much then for the introduction. So I would like to talk with you about visual awareness. And the way that I'm going to do this, hey, this looks different to what I'm used to. So think of this as one number, and so number 127. And I'm going to do a couple of experiments with you. And so maybe you want to remember this number for later. And the second experiment is very simple. I'm going to present a visual stimulus around here. And so this is an experiment about consciousness. So if you see it and your consciousness of it, please raise your hand. If you don't see it, don't. So it's a very simple task. 1, 2, 3. Okay, yeah, many of you are conscious. I'm glad. Now I'm going to try it again. 1, 2, 3. Now I'm going to make it very difficult. So this is for the careful viewers. 1, 2, 3. Nobody saw it? Okay. Let's try again. 1, 2, 3. Yeah, some people saw it. Okay, I'll be honest with you. The second one, there was no stimulus. And so I tried to kind of lure you into a false alarm. It didn't work. But even if it happened, you don't need to be ashamed. So because for each and every one of us, there is, of course, a contrast level where it becomes very difficult to see. And that's a good trick. If you want to study consciousness, then you can present physically the same stimulus. And in some trials, people report it, so it entered into the consciousness. In some cases, they won't report it, so it did not enter into the consciousness. And you can compare what happens in the brain and compare the two situations, which is what we set out to do in an experiment. And before I'm going to show the results, there's a theory that later turned out to give quite a good account on what we found. The signal detection theory. I guess many of you may have heard of it. It became famous, I think, in the 60s. There is 2 scientists that are quite attached to it. It's Green and Svets, but there have been other people working on this. And the idea is the following. It's a very simple idea. If you present a weak stimulus, then there will be an internal representation of that stimulus in the brain that is here modeled by a green Gaussian. And so if you then present on a particular trial the stimulus, you sample from this distribution. So you might have a particular internal strength. And then in the signal detection theory, there's a threshold. And if the internal strength is above the threshold, you're going to say yes, and we call it a hit. But it may happen because of maybe the stochasticity in your nervous system that you say no, even though there was a stimulus, because it stays below the threshold, and it would then be a miss. Now, if there's no stimulus, there's another distribution here in black. And so typically you will stay below the threshold, and that would be called a correct rejection. But again, due to the noise in the system, maybe sometimes you say, I saw a stimulus, even though there was no stimulus. That would be called a false alarm. Now, if the stimulus on average is much stronger, then you get, of course, a shift of the Gaussian distribution to the right. And if you then apply the same threshold, you see that you have still quite a number of false alarms. Maybe it's a good idea to raise the threshold to have fewer false alarms. But and with a strong stimulus, you can afford to do so because you don't have so many misses. But if you would apply this high threshold to a weak stimulus, then the part of the green distribution to the left of this red line is pretty large. So you're going to have quite a number of misses. So there's this sort of trade-off between where you want to put the threshold and how many misses and how many false alarms you're going to get. And it depends really on the task. So if you're working on an airport and you are checking luggage, you might want to put the threshold very low because the consequences of a miss are much more severe then consequence of the false alarm, right? False alarm is just opening the suitcase and doing another check. Okay, so this is all quite well described by the signal detection theory. And in this study, we asked a couple of questions. So where is the signal in the brain, this internal representation of strength? What causes the variability, basically the width of this distribution? And what is this threshold? What does it correspond to in the brain? And when we then got the data, we started to support a theory that had been published by Stan many years before. And so the theory is the following, that if you present a stimulus to the brain, it of course enters into the eye, the LGN, and then goes to the back where primary visual cortex is, and then it's propagated to the rest of the brain into frontal areas. And here Stan predicted that there's a process that we now know as ignition, and that gives rise to a self-stabilized pattern through these recurrent connections, such that if you take the stimulus away, there's still a trace that the stimulus was there. You can call it a working memory of the stimulus that you just saw. Now, the prediction would be that if you don't see the stimulus, the feed-forward propagation might not be efficient enough. Ignition may not happen. So if there's no ignition, there's going to be a miss. And if there is ignition, there's going to be a hit. That's the whole idea behind it. Okay. So we trained monkeys on the task, basically the same as you've been doing, with a small modification. There was always this small dot on the screen. And if the monkeys on those trials, 50% of trials without a stimulus, they wanted to report that they hadn't seen anything, they could always make an eye movement to this small dot, reject dot we call it. But then on the trials in which there was a stimulus, then after a delay, the monkey made an eye movement to the position where we had presented the stimulus, reporting, I saw something. Now the behavior is very straightforward. If the stimulus is strong, high contrast, the animal basically always reports it, makes an eye movement to it. Then if you decrease the contrast, of course, the animal is going to have misses, and at some point, performance becomes very poor. Now for the analysis, we defined two thresholds, one at 80%, one at 40%, and we said if accuracy is above 80%, then it's an easy stimulus. If it's below 40%, it's a difficult stimulus, and intermediate if it's in between, just for the analysis. And we recorded neural activity across many structures, or actually 3 structures: primary visual cortex, and before, which is a higher level visual area, and also in prefrontal cortex. Now I should say that here there are neurons that code for eye movements, and we are not going to dissociate in this study the eye movement plan from the pure conscious percept. So that's sort of a limitation of this study. And now for the results, and when we started to do this task, we were in for a surprise. And here I'm showing you the activity listed by the difficult, low contrast stimuli, intermediate, and easy stimuli. And here we're starting trials based on whether the animal reported the stimulus or not. And we made sure that in this comparison, the contrast was the same. Okay, so you see that on those trials in which the animal is going to report the stimulus on a hit, the activity was higher than on the misses, but the stimulus was the same. That was quite surprising to me. So there's some variability in the quality of signal transfer between the eye and primary visual cortex. These are these low-level visual pathways. I thought they should be more reliable, but they're not. And another thing that you could note is the easy miss trials, when the animal fails to see a stimulus, that was actually quite apparent. Activity in V1 is stronger than on the difficult hit trials, right here in green. But this means that if a stimulus reaches a certain minimum level of activity in V1, there's no guarantee that it will be reported, as if it's still possible that there's a lot of V1 activity, but it gets lost downstream maybe on its way to ignition. Now then we also recorded from V4, which is a higher visual area, and we basically got the same result. Also here, a certain level of activity here on the miss trials was no guarantee for conscious perception. So activity here is at least as strong as on the difficult hit trials. Okay, so even a certain level of activity in V4 is no guarantee that the stimulus will reach conscious perception. Now in the dorsolateral prefrontal cortex, we got something that was qualitatively different. So here we get this bistable behavior. Whenever the animal is going to report, there is this high level of activity. And whenever he's going to miss, there's a low level of activity. And you see on the easy miss trials, there is actually some ramping, but apparently not enough to cause a self-sustained activity pattern, because when the stimulus is no longer there, the activity fades away again. So if there's one area where you could put a threshold between hits and misses, then it's in frontal cortex. Now we also looked at those trials in which there was no stimulus. And here in black, you see the activity pattern on the correct rejections. So the animal didn't see a stimulus and reported not seeing a stimulus. But in blue, it's interesting. These are the missed trials. And you see that activity ramps to the same level as on the when there was a stimulus that was reported. So here, of course, he's also going to make an eye movement. That's the caveat here. We're not dissociating eye movements from the pure conscious percept, but it's intriguing, right? So, and so if there's one area that conforms to this prediction of the signal detection theory, then it's definitely going to be dorsolateral prefrontal cortex, because they nicely sort according to the predictions. So weak activity on the correct rejections, intermediate on the false alarms and the misses, and the strongest activity on the hits. And you see towards the end actually becomes completely predictive of what the animal is going to report. Now you see the slow ramp, and we cannot prove that in the data, but we actually suspect that on the trials with false alarms, there is a low level of activity, and there are then spontaneous, we think, faster ramps, like a bistable system. But if they happen at different time points relative to time zero, and you average many of those trials, then it looks like a slow ramp. But again, we cannot really prove that. Okay, so I showed these data to Stan maybe 7 or 8 years back, and Stan became very excited. He said, that's exactly what I predicted. And this is a modeling effort by him and Pierre Jean-Jeu and years back. And he said, you know what I'm going to do? I'm going to make you the simplest model that you ever saw, and that can account for the data. And this is the model. The model models the whole brain with only 5 neurons in total. Okay. So there's 1 neuron for the LGN, 1 neuron for V1, 1 neuron for V4, 1 from parietal cortex, frontal cortex. You see there are feed-forward connections, there are feedback connections, and there are self-connections. And the model works as follows. So activity comes in the LGN, gets propagated through the feed-forward connections. Now, the connections between parietal and frontal cortex and the self-connections here are made so strong that if an activity reaches a certain level, it can self-sustain. So if that happens, that we call ignition. And if you not take the stimulus away, well, activity can self-sustain. So the model can later then report it saw something, that it would be a hit. Now, on the trials that are misses, you can present the same stimulus, but due to the stochasticity of all these neurons, it can happen that the activity just stays below the threshold for self-sustain. If you then take the stimulus away, then activity collapses also in the rest of the network. And it would be a miss. And basically, this model replicates many of the aspects in the data that are presented to you. So this, of course, is difference between hits and misses that also happen at early levels, because if there is just due to the stochasticity in early levels, there's more activity, there's more probability that this will cascade and also cause ignition in frontal cortex. And you see in frontal cortex itself, there is more this bi-stable activity pattern. And on the misses, you see this ramp, but it apparently is not enough on those trials where there will be a miss to cause self-sustained activity. You also get the same effects on the false alarms and on the correct rejections. And in the model, we can check what happens on individual trials because you have the whole model in your hand. And then you indeed see that on those trials, there are actually quite abrupt ignitions. But if you then average across many trials, they become like a slow ramp. Okay. So this is the modeling effort. I think Stan did this on a rainy Sunday afternoon, and I feel a bit embarrassed because Stan is here and I told this joke maybe already 5 times. But this is the efforts of 3 PhD students in the lab who worked hard on this for 4 years. Okay, so I know in your beautiful institutes, you're doing theory and you're doing experiment. And if you're kind of can choose, maybe modeling is something that you can do very efficiently and get a lot of output. But of course, the data itself is also highly valuable. Okay, so conclusions of this first part. I think it's evident that the variations in internal signal strength are well explained by the variation in the efficacy of the forward propagation. We see that the threshold for perception might correspond for the threshold of the self-sustained activity, some bifurcation in the network. And also that the false alarms are well explained by the spontaneous ignitions due to the stochasticity in the network. Sometimes it happens that the activity becomes self-sustained. It only happens in the monkeys and also in the model, a very small percentage of trials, 3% of trials or so. Okay. So this supports this idea that there's a special role for frontal cortex for causing ignition. But now I actually should admit that for me, it's definitely not the full story. So I don't think that frontal cortex has a unique and very special role in causing ignition. So here you get the idea that the important stuff happens in frontal cortex, but there are situations actually where you also need involvement of visual cortex to produce conscious perception. And I'll give you one example. So what I'm going to do, and I'm not sure how well this is going to work because the timing is not completely predictable, but have a look at the picture that I'm going to present to you very briefly and try to see what is there. Okay, are you ready? 1, 2, 3. Did you see it? So what was there? Leaves, wood. I hear a murmur, but not no solution. So any other thing you saw? Rocks, yeah. Okay. Here it is. Okay, so what happens here is you probably saw colors, textures, but of course here you see a bird that is also somewhat camouflaged. And this is one of these situations where your visual brain needs time to put together the fragments into a coherent whole. Okay, and actually we now have evidence that to get conscious perception of these figure ground percepts, you really have a strong involvement also of visual cortex. So if you think about the neural processes, maybe you could call it the global neuronal workspace as Stan does, where there are these different processors and they have to work together to get conscious percept. I would like to argue that in some cases, even visual cortex, maybe even primary visual cortex, becomes part of the global neuronal workspace for conscious perception. Okay? And actually what seems to happen, and we have many experiments in which we demonstrated that, is that all those image elements that are bound in your perception, they are labeled with an enhanced response, and psychologists call this object-based attention. Attention is directed to all the features that belong to a single perceptual object. Now, then again, as I said in the beginning, in the consciousness literature, people can debate endlessly about it, and not everybody will agree with me, but I think this is what many people call access awareness. So the things that you're seeing and attending, and the things that are also there, but that you're not attending, but that you could attend to if you wanted to, are typically called phenomenal awareness. Okay, now if you would shift your attention from the bird to the stick, right, then the roles switch. Now the stick is in access awareness, and the bird, well, might actually disintegrate as an object. It's no longer in access awareness. Okay, you could, of course, direct your attention and then reconstruct the bird, but at that point, it's no longer in access awareness. Okay, now I want to show you some data of a bird-like task, but much simpler. So this is a figure ground task in which you see some elementary features that form a figure on the background of the same elementary features, but of a different orientation. And a very good colleague and friend of mine, Victor Lamme, has studied this quite a lot. And this is from a paper also more than 25 years old. And this is recordings from primary visual cortex. So you see here's the receptive field, falls on some figural elements, and here the same elements are there, but they're part of the background. And if you then look at the neural responses in primary visual cortex, what you see is you have this first phase, this feed-forward phase, where activity comes from the eye, starts to elicit the response. Then V1, of course, sends the information to higher visual areas, and they start to feed back. And then you get an extra activity level if the image elements in the receptive field belong to a figure as opposed to the background. Okay, so basically in the beginning, the neural responses are driven by what's going on in the receptive field. It's the same for figure and ground, and then when the feedback comes, there's more activity on those trials where the figure happens to fall in the receptive field. And Victor, also in a somewhat older paper, demonstrated that this is also correlated with conscious perception. So he showed these figure ground stimuli to monkeys, and typically the animals saw that there was a figure and reported it with an eye movement. And on those trials, there was a clear difference. So this labeling process I was talking about between figure and ground. But there were also a few trials in which the animal failed to perceive the figure. And you see in these trials, the feed-forward response was intact, but the figure-ground modulation, the difference between figure and ground, did not happen. Okay, so this is, of course, in some sense, correlative evidence. So we would like to have some causal evidence. So we wanted to do optogenetics. And so these are the figure ground responses in the macaque monkey. You see that figures elicit more activity than the background. Actually, you get the same effect in humans. So we sometimes record from the visual cortex of humans in a collaboration with neurosurgeons, get the same effect. But for our optogenetic study, it was very important to replicate the effect also in mice. So also in mice, you see that figures elicit more activity than the background. Okay, so now we can then train mice to do figure ground perception. So they're looking at a figure stimulus, figure either on the left or figure either on the right. And then the mouse is supposed to lick on the double-sided lick spout on the side where he sees the stimulus. And then we use optogenetics. This is the work that was carried out by Lisa Kirchberger. And so on those trials, here you see the figure ground response. You can just knock out, using optogenetic trick, optogenetic silencer, knock out all the spikes from primary visual cortex, and then see what the consequences are for perception. Now, optogenetics is a temporally very precise method. So what we can do is we can shift the timing of the light to let through only the feed-forward response and selectively knock down the late effect, right, where you see this figure ground modulation. Or also let a little bit of this figure ground modulation through, or even more, and see what happens to the perception of the mouse. And this is what we found. So the mouse was either doing a figure ground stimulus, but also he had a condition in which he just needed to detect contrast on the left or contrast on the right, basically what you were doing in the beginning of my talk. Now, what happened if we did optogenetic silencing and we knocked out the entire visual response? The mouse was basically blind. So he could not do contrast detection, he could not do figure grounds. But if he then let through only the feed-forward response and only knocked down the late effect, then the mouse could do contrast perception, but he was still miserable at figure ground. He couldn't do it. So we really had to let through some of this late V1 activity, so when the feedback effects come in, and to kind of have the mouse report that there was a figure ground perception. Okay. So we also know from previous work that this figure ground effect is much stronger if the animal is interested in the figure, so if he pays attention to it. And therefore, I think this is pretty strong evidence that if you think about the global neural network space, that actually it reaches back to primary visual cortex if a figure ground segregation is required. You really need to do this labeling process, labeling all those image elements to produce this coherent percept. And that is something where early visual cortex really is instrumental, because there all these individual elements are represented with sufficient resolution. Okay, so what I kind of told you so far kind of can be summarized as follows. So there are things that you perceive and attend. So that is what philosophers of consciousness call access awareness. Then there are some items that you could attend if you wanted to, but you're not attending yet. They are part of phenomenal awareness. And then there is items that you just fail to perceive. So you could not even see them if you wanted to, but they're outside awareness. And now I would like to make a parallel to memory. So in the beginning of my talk, I gave you a number. Do you remember the number? 127. So now, crucial question, where was this number all this time, right? Where was it in your brain? For me, an important question. And to be honest, I'm not going to answer it for you, but I would also like to know. And I would like to argue that in memory, you make similar manipulations. So there are things in your memory that you're attending. So number 129, when it produced the 3 axes, it probably came into your working memory and you were attending it. But there are other things that are to some degree in your memory, but you're not attending them. So one example is iconic memory. So if I present you a stimulus very briefly, there's a brief time that it doesn't fade away yet. That's what psychologists call iconic memory. I don't have time to really go into the details of that. And then of those things that you briefly see, you can grasp a couple of things to remember and to report. Another type of what I would like to consider a type of memory that's part of phenomenal awareness is accessory memory. And I will give you one example of an experiment where we think we have an angle on accessory memories. And of course, if you forgot the number, then it's lost, right? So I think the parallel between perception and memory is quite defendable. Okay, now the task in which you see this transition between access consciousness and phenomenal awareness in memory. So to test this, and this is actually an experiment that was carried out by Doris Dijksterhuis, also in collaboration with Stan de Heen, we present to the human subjects a sentence, Barack Obama is one word, and the subject sees that on the screen, and then the word and follows, and then the word, there's one word, Jennifer Aniston, set in a bar. Then there's a pronoun, he or she, ordered a beer. Okay, and during this task, we have a collaboration with neurosurgeons who implant electrodes in the brains of people with epilepsy. And through these electrodes, we can shift microwires that allow us to record these sort of Jennifer Aniston cells, right? You probably have heard of it. It was first done by Isaac Fried and his collaborators. And so we can then record from cells that respond to specific individuals. And it turns out that some of these cells also respond if you present these individuals as a word. And this shows you then the activity. This is actually decoder analysis, but you can think of it as Barack Obama being presented in the sentence. You see Barack can be presented as the first noun, or in another sentence, Barack would appear as the second noun. And you see that when you present Barack, it's very active, and then there's a little bit of lingering activity, but it's definitely not as strong as when you present Barack at the first time. Okay, and then becomes interesting because now there's the pronoun. Now, if the pronoun is he refers to Barack. You see a reactivation of the concept cells coding for Barack Obama. Okay, so what happened here? Barack was briefly there, maybe then Jennifer came along, so Barack was in access awareness in your memory. You were thinking about it actively. Then it was still in memory, but Jennifer came along, right? So maybe it now become part of accessory memory. Right, so you might call phenomenal awareness. But then the pronoun comes, and if the pronoun refers to the concept, then the concept comes back into access awareness. Right, so you see this interplay between access awareness and phenomenal awareness. Phenomenal awareness providing a context for the activation of the right concepts in access awareness. So I think that it's a nice idea to generalize these concepts to memory operations. And this is another rendering of the global neuronal workspace theory. So I have 5 minutes. I move on a little bit to prosthesis. And basically in our lab, we have been implanting electrode arrays in the visual cortex of monkeys, and we were doing hundreds of electrodes. And at some point in the lab, we realized maybe we should ramp it up a little bit more. So let's go to 1,000 electrodes and see if we directly can impose images onto the visual cortex. And that could be a relief for some of the people, 40 million people in the world who are blind. And in our work, we are now focusing on primary visual cortex, but you could, of course, also consider the eye or the LGN. And we know that if you stimulate a cell in primary visual cortex, we're using these Utah electrode arrays, that somebody who has been blind for several years sees a dot of light. And the visual cortex is a map of space, right? And if you stimulate a particular location in the map, this person will see a dot of light at the corresponding location in the visual field. Okay, so that's what happens. We call this a phosphene. If you have 1,000 electrodes, you can produce 1,000 phosphenes. And the idea is to activate some of them, and then you can write basically images directly to the visual cortex. And like a matrix board, right? If you flash one bulb, the person will see a dot, but if you flash the right pattern, you can see something that is more meaningful. And this is what such a system will look like. So you have a camera, and you need a processor that changes camera images into brain stimulation patterns, maybe a bit more abstract than the full image. And you need to send it wirelessly to the electrodes in the brain. So that's what we set out to do. So we implanted 16 of these 64 channel Utah arrays in the visual cortex. This is what the implant looks like. And this shows you schematically where we implanted these 16 Utah electrode arrays. So 14 of them were actually in primary visual cortex. These are those. We can map the receptive fields, know where we can then produce the phosphenes. And we did this for 2 monkeys. Now I want to show you one movie. This works. Do we have to change this or? Let me see, how should I start up the movie? There's something I can do with the pointer?
Get rid of the pointer.
Where do I do that?
Lower left.
Here? This one?
Come back to the arrow.
Start it. You know how to do it? Okay. This works. This will work. It's fine. Oh no, you don't see it here. Okay, thanks. Thanks a lot. Okay, let's see where we can now start up the movie. Still not. Oh, now it's the movie. Okay, so basically you see the arrays. Of course, this doesn't happen in the lab. You then don't see it through the skull. You see there's a moving bar, and you see that there's a wave of activity across the cortex. Right, and the moment that these cells become active allow us to precisely map where the receptive fields are of all these neurons. Now we are not really super interested in recording. We're more interested in stimulation. So we are now stimulating one of the electrodes, and we're recording the eye movements of the monkey. We trained the monkey to make an eye movement to where he sees something. And you see this works. So the monkey is making an eye movement approximately in the direction of where we stimulate the cortex. So reproducing something that other labs had shown before us, but now for hundreds of electrodes. But the real important question for us was this matrix board analogy. Can we really impose patterns onto the visual cortex that are recognizable? So we trained the monkeys in a task in which they had to recognize letters. So here the monkey saw letter D, and then we hoped, and then there was a choice menu, and we hoped that the monkey then would report the corresponding letter. We trained, of course, extensively on this task. And in the most exciting part of the experiment, we did not show the letter anymore, but we actually electrically activated a couple of neurons, and then hoping that the animal would report the letter that we were trying to impose on the cortex. And importantly, that worked. So here we're trying to write the letter L, and you see the monkey makes an eye movement to the letter L. Letter O, letter A. So this was very, very exciting for us. So when we got this result, we were very happy and made a small dent in the lab. Okay, so for us, we are not there. The system that will need to be developed is not yet there. It's nice to see the results of an experiment that was mainly done in the lab of Eduardo Fernandez in collaboration with us, but they really did most of the work, where they implanted a human also with a Utah electrode array in the primary visual cortex. So this shows you approximately where the electrode was implanted. And when they then activated a set of 16 electrodes in the shape of a large square, she reported seeing a letter O. When they did only 4 electrodes neighboring, she reported a small letter O. So that was, of course, very exciting and what we hoped for. Now here she reported the letter I. Okay. But here she reported the letter L. So this also demonstrates to you that we don't have full control of what she's going to perceive. I think this is related to the fact that these arrays are very small, only 4 by 4 millimeters. So therefore, there is probably on that small scale, there's going to be some interference patterns. And I hope this will work.
Fue indescriptible y bueno, yo estaba pletórica y bueno, y todo el equipo también, entonces claro, os decía, pues estoy viendo un punto luminoso y todo el mundo calla, pero chicos, ¿qué estoy diciendo? Que estoy viendo un punto. Y bueno, nada, al final pues fue una explosión de alegría y todos ahí contentísimos. Pero por supuesto que si ahora hubiese uno que fuese inalámbrico, que se pudiera trabajar con mucho más electrodos y se pudiera ver, yo ya no pido una visión, simplemente desprender una puerta, un obstáculo, lo volvería a hacer, sí, sin duda alguna, sí. Fue indefinible.
Okay, so of course you should realize this is an experiment, right? It's not a treatment. And so Eduardo spent a lot of time explaining to the blind people who are part of his experiments that they should not expect to see again. But if you see the degree of joy, just by seeing a dot of light, I think it's quite touching to see that. But we should also explain to them that this is still very, very limited. So one limitation is that it only works for about half a year in our lab. After half a year to a year, then the electrodes stop working because there's encapsulation gliosis, and that causes a loss of function. So something that I think there are ideas how to solve this, but it still needs to be proven. And the other thing is the degree of coverage of the visual field. So if you put one Utah electrode array, 4 by 4 millimeters, in primary visual cortex, which has a surface area of 25 square centimeters on the left and another 25 square centimeters on the right in humans, then you should realize that you're covering less than 1% of the visual field. So that's another challenge to make a prosthesis that will target a sufficient surface area of primary visual cortex. Okay, and I look at Alain, I think it's probably already a little bit over time. So here are the people who did the work. So here are my mouse is not visible anymore, but in the top row, the people who did the work for the visual consciousness project. The second row, the people who did the figure groundwork. Third row, the people who did the language study on consciousness. And the lower row, people who are involved in the visual prosthesis work. Thank you very much.
Thank you.